we are going to be talking about teaching the science practices, effective engagement in the AP course. So if you don't teach AP, that's also okay. You can teach the science practices in all of the science courses. So there'll be tips and ideas regardless, but these are specifically for the science practices that are in the CED from the College Board. So hopefully everybody's in the right place. Text your friends if you're afraid they're somewhere else. You're welcome to come in late if they want to. If you know of anybody else who's teaching AP Bio. That's okay. All right. Anyway, welcome. My name is Paula Phillips, and I came here from upstate New York. And I've been living in upstate New York now for 7 years, teaching in a really, really rural, very small public school. Before that, I was teaching in Florida at a really, really large independent school. And before that, I was teaching in rural North Carolina, also in public school. So I've been teaching since 1992, and I've been teaching AP biology since then. And I have had the privilege of working in so many venues associated with AP. I work for the College Board and Educational Testing Services, and I work with BFW, who very kindly brought me here today to talk to you guys about the science practices. I worked most recently with these other two wonderful individuals, Jim Smanik and Gordon Uno, on the Teacher's Edition for their textbook, which is biology for the AP biology course. And our real role in that book was adding in all of the material that goes around in the ancillary locations on the textbook to give ideas for how you can incorporate the content that was in the reading part of the text with the science practice skills. So there's all kinds of things that you can interact with on that, and I'm going to be talking about a couple of them today. So if anybody's interested in getting the presentation, I'm happy to share it with you. There'll be, if you leave your email address here, we can get it to you, or there's also going to be the name of somebody at BFW that you can send, can ask for it as well. I'm kind of a walker. I'm not really one to stand behind a podium. This doesn't feel very natural for me, so I'm going to be moving around. I'm finding this odd. Can you hear me okay if I go back and forth from this? Okay. All right. We're going to start out by just thinking about, what are the science practices that are utilized in biology? Like, what are the kinds of things besides the content that you want your students to be able to do at the end of your course? So if you're teaching a bunch of students in AP biology, what do you want them to be able to do with the biology content that they're learning? What are some of the things that, that you can think of? Calculate, analyze, graph, question, apply, identify. I'm noticing a theme of these are the task verbs for the College Board for their science practice skills. And one of the things that I really want to highlight about the science practice skills is they are skills. And like every other skill that we ever learn to do, you really have to practice them, right? So thinking about ways that we can incorporate those skills into our everyday lesson plans rather than thinking of them more as like planning for them once a week or planning for them twice a week. How can we incorporate them into our lesson plans every day? So for some of you, you may be teaching at a school where you have 120 minutes a day with your students. That's not how it is at my school. We have 44-minute class periods, and we meet with them once a day. And so really thinking about what is the best way that I can engage with the students in the classroom. That's kind of what we're going to be. Looking at, and thinking of how can I do it every day. So on the far right here are the science practice skills. In case if anybody wants to just a reminder of science practice skills, I've listed them. This is straight off of the CED. But we're asking the students to do concepts and explanation, visual representations, one of my favorites, questions and methods, representing and describing data, statistical test data analysis, and argumentation. And I would say that for me, when the CED first came out, one of my goals was to consider how I can mindfully incorporate one or more of these science practice skills for the students every single day, every single lesson, so that it's not just me standing in the front teaching for an entire period. Regardless of what content we're covering, every day I do something, whether it's 2 minutes, 5 minutes, 10 minutes, the whole period. And so that's kind of what we're going to look at here. And the reason that I think that's important, well, I think that's important for a lot of reasons. But one of the reasons that that's important is if we think sort of with the end in mind, it'd be lovely if every, kid in the school system took science just for the love of science. That would be awesome. But for this particular course, the students are always thinking about the AP exam. Like, how am I preparing for the AP exam? And that's how I, as a teacher, also feel. I want them to be successful. I want them to get the credit from the AP exam. And I want them to feel moving forward like they're getting something every single day and they're building on what they already know. So with the end in mind, I started thinking about, what are the FRQs? What are the students being asked to do by the College Board, when they are taking the exam? So this is the one from 2023. If you taught last year, you've already seen these. If you didn't teach last year, you also may have seen these. But I just want to pull up the first question. Scrolling. Okay. This is really a typical format of the questions where the students are provided a long stimulus, because there's no way we can teach them every little bit of content, every example of an investigation that they may ever see in their lives. So they get a lot of stimulus with a background of information that the students have to be able to incorporate content-wise. So they've got a lot of content here. And then they're presented with a portion of the stimulus that is either a graphical representation of data, it could be a data table. In this case, they're looking at a visual representation of a metabolic pathway. So the students are reading this background information that probably they've never seen before, and then they're looking at an illustration. One thing I always tell my students is read the caption on the figure, whatever that is. So we're looking at a model of the regulation of expression of a gene, and then scrolling. Then they give, typically in question number 1, they're going to give even more information. So they've got a data table here. So this is visual representation. So this is science practice 4. Sorry, I meant visual. The first visual image was science practice 2. This one is science practice 4. And then what are we asking the students to do with that? So I know you're familiar already with all of these task verbs because you were already saying them. But can you describe something? Can you explain something? Can you identify a dependent variable? Justify the use of a control? Can you identify from the data table something particular about the data? Hey, what if we did something different? Can you predict the effects that this difference would have, and then justify your prediction? So, you can see in this one question, all of the students are being asked to really demonstrate an understanding of all of these science practice skills. And my goal then every single day is to help the students achieve this, right? So I'm not always giving them a whole FRQ, but I am really making sure that we're practicing all of those things. So So what might that look like if I were making out my lesson plan? Like, what is that going to look like? How can we provide these little daily opportunities for the students? So, I mean, it's a variety of things, right? I'm not doing the same thing every day. That doesn't make sense for the content, and it would certainly get tiring. But, you know, the first thing is probably the easiest, and that is access to investigations. Now, you know that the College Board, you know, you all have access to the College Board's lab manual. It's freely available online. It has a link to it in the CED that's online. So there's 13 labs right there that you can choose to do. And I do some of them. But just providing access to investigations. And when you're talking to the students about the investigations, giving them an opportunity to talk about the components of the investigation. Things like what's the null hypothesis versus the alternative hypothesis. You know, which of these things that are in this investigation would be the negative control or the positive control? Why do we have to have a control at all? These are the kinds of things that the students might need to be able to articulate themselves on the AP exam. And the wording of that is very difficult for a 16, 17, 18-year-old kid. Right, so providing them opportunities to actually talk about those components in their own words so they can write them down in their own words if they're asked to do that. Science warm-ups, I love these. Class starters, you might have called them in elementary school. I think we used to call them, oh my gosh, what do we call those little things that were warm-ups? Bell ringers, right, bell ringers, pop-up questions. These I love. These are like 2 minutes, 3 minutes, any day. You can pop something up on the screen while you're taking attendance. You can integrate it halfway through your lesson, so you've done a bunch of content and you're like, did these kids get anything that I just said? Then you can give them 2 or 3 minutes to practice something in the middle, or you can use it at the end as some kind of exit ticket. This one is really critical, I think, for being able to get at Science Practice 6. You've just taught a big lesson on, with some, really critical content like how enzymes control a metabolic pathway. And now what you want to know is, did you understand that well enough to say what would happen if the enzymes weren't functioning properly? What if enzyme number 1 in the metabolic pathway isn't functioning properly? What do you predict would happen then? So these are this what if questions, and then asking the students to make a claim about what they think would happen, what is their evidence to support that, and what is their reasoning. So we're doing a little CER there. And then justifying their predictions. I think that's critical, and I have some examples coming up of how to do that. I have my students all the time write 1-minute essays, 2-minute essays, some kind of quick write. My students are asked to keep a notebook. It's like a portfolio. They put all of their lab materials in there, but also whenever we do something like this, which is daily, I have them put the date and then their answer. So they keep them. And that's twofold for me. One is I don't want to grade those every day, right? If it's something that they need feedback on, I want to give them the feedback, but I don't want to give them a grade on it every single day. But I do want them to have access to it throughout the year. It does 2 things. 1, it keeps all their stuff in one place, but 2, it shows them their own progression throughout the year, which I think is critical. Because when the kids come in this class, it's likely the first time they've experienced the uncomfortableness of not knowing an answer to something. They come in and they're used to being there, hand up, and somehow in AP biology, they're silent sometimes at the beginning. So it helps them to see their progression in the class, which I think is a powerful thing for the students. Translate this, picture this, narrate this. Hey, can you explain this in your own words to your seatmate? Gallery walks, misconception checks. These are all things that we are going to be looking at. Examples, that's what I have sprinkled into this presentation. But how can we use them to inform our teaching? Well, number 1 in Ms. Phillips' classroom, they don't need to be scored. Right, these are things that we want the students to have feedback on, but I don't necessarily want to put them in the grade book. So the students can review them in small groups. They could lead a class discussion or an activity. They could be used as an exit ticket, so you could take them up as the students are leaving class. But it helps the students focus on demonstrating what they can do with what you've just gone over in the classroom. So you're not taking 3 or 4 days between like giving them this really heavy content, and then there's more really heavy content after that, and then there's this really long lab that the students really need to be able to, like link back to that content. And it's a lot for students. It's a lot for adults, but it's a lot for students. And so every single day, like helping them make these connections like this, I think is pretty critical. So we're going to start with Science Practice 1, and I'm going to go through a couple things for each of the science practices. So Science Practice 1 is, can you describe and explain? And the first thing here is, every year on the AP exam, the students are asked to describe something and they are asked to explain something. And I don't know about your students, but my students don't know the difference sometimes, right? They're not really sure what the difference is. So that's day number 1, right? Like first thing, straight out the gate, let's talk about what are these task verbs intending for you to do. So for my students, I give them kind of a silly explanation for a description. I work in a really small school. Everybody knows me at the school because it's a tiny little town, and they know I don't know anything about the sports balls, right? Like I don't know baseball, I don't know basketball, I don't know football, I know none of that. And so I always use a sports ball analogy, right? So a description is something that in the sports ball, an announcer might be standing with a microphone talking about. A player with the blue t-shirt just stepped onto home plate. They're hitting at a ball that was just thrown at them by the pitcher. Oh, they made contact. Now they're running to the first base. Right? They're not really explaining the game of baseball. They're saying what's happening in the moment. That's a description, right? Versus an explanation where they're pulling pieces together. Right, an explanation is, how, why, when, where, but like pulling it all together. And we'll look at some examples, and I'll give you an opportunity to think like, how would I answer that if I were asked to describe it? Versus how would I answer that if I were asked to explain it? Because it's a little bit different, right? Like those two. And honestly, if we just imagine that the students know that from the beginning, I can tell you as a reader of 30 years on the AP exam, they don't know it. If you don't tell them, they don't know, right? They will give a really lengthy explanation at a time when they really only needed a 1-sentence description, or they'll give a tiny little description when they were asked to make an explanation. So let's develop parts of a lesson plan that are going to help the students make that distinction. So here's a strategy. Ask a summative type question. Now, I pluck summative questions off of old FRQs all the time, like screen snap, screen snap the questions. So these are straight off of an old AP exam, not even that old, this is just from a couple of years ago. So these are some examples. Describe the characteristics of the plasma membrane that prevent simple diffusion of these ions across the membrane. Right, here is a really good example of a can you describe something? Right, so you can actually use this one pretty early in the school year because this goes with Unit 2, where the students are learning about the cell membrane. But describing the characteristics of the plasma membrane is what we're looking at here, right? So if your students sat down, what would they say? What do you think your students would write, or what would you write? Describe the characteristics of the plasma membrane that prevent ions from moving across. Item number 1, this is relating it to the content. They need to know what the plasma membrane is made out of, right? Like what is the plasma membrane made out of? So the students here should be talking about the phospholipids have tails that are hydrophobic, and these ions can't move through that hydrophobic layer. Done, they're finished. That is the answer to this question. Describe the characteristics of the plasma membrane that stop this motion. That's it. That's what they've done. The next question is an explain question. Explain why ATP is required for the activity of sodium potassium ATPase. Like why do you have to have ATP for that? In this one, again, it's pulling back content, but here the students have to say an explanation of that relates the movement of sodium and potassium with the phospholipid bilayer and the use of this enzyme. So here, what might you expect them to say? They've got to say something about the movement of these ions against their concentration gradient, right? They're moving against their concentration gradient. So why do we have ATPase involved? Because what do they need to do to the ATP? This is going to require energy because it's active transport. They need to break the last phosphate group off of the ATP, and that's what's going to provide the energy. They could talk about energy coupling if they wanted to, but this is a much more involved question than the question that's here. All right, the next one. Man, I love this one. I don't know what it is about this one that I like so much. Unit 3 is really hard for students if you remember the units in order. Unit 3 is the one that goes over photosynthesis, cellular respiration, enzymes. There's a lot. Unit 3 is the longest. It has the most components of any of the other units, really. But this is another strategy of ask a summative type question and then follow it up with the misconception check, or a 1-minute essay or a 2-minute essay. I guess it depends on whether they got it right or not, like what you want to move forward with. But this is a really good example of a question that gets at a super common misconception for the students. So the question is, a worker at a fast food restaurant famously lost a lot of weight eating a low-calorie diet. Which of the following best describes what happened to the individual's mass? So this happens to be a multiple choice question. It wouldn't have to be. You could give it as a free response question where they would have to write out the answer. But the answer that we're looking for here is rarely the answer that most students are able to choose the first time through. So, for example, the correct answer is the first one, A. Right? The correct answer is A. The mass is released as carbon dioxide and water. All three of the distractor responses here get at the same misconception, which is that students have difficulty thinking about matter and energy as two separate things. They want to imagine that they are the same, that one of them is convertible to the other one, and that is not the case. So this is a really good example of a question that's like really quick. You could do it right at the beginning of class, you could do it halfway through class, you could do it at the end of class. Ask them the question, have them choose the answer. You could have them turn and talk to a neighbor if you wanted to, but then if they've missed it, give them the misconception check. Like, this is a common misconception, which is that matter and energy are interchangeable, and that is not true. So there's a good example of that. You could ask them, hey, write for as many things as you can in 1 minute that illustrate how this is not the case, how the misconception is not the case. This is another one that I love. This is a great example of a lab investigation. I don't know how many of you have a limited budget at your school. I do. I have an extremely limited budget. I'm at a super tiny school that is very rural. I think last year I had like $100 to spend for the entire school year. Don't feel sorry for me, because I love finding ways to integrate data collection opportunities that are free. Not only are free, but require very little setup on the part of Ms. Phillips, because I was hearing some of you guys talking earlier, you've got a bunch of preps, and like anybody in a small school, I also have several different preps. This is a great lab, but you don't have to do the lab. You can just give the students this image. So analyze components of an investigation. You could talk about this as an investigation and ask the students, what are we using here as a positive control, or what is the negative control? Or you can perform the investigation, you can do it as a misconception check, but this question is phenomenal. 3 identical plates of radish seeds are incubated under 3 different conditions with the results as shown. So light with no water, light and water, no light and water. And then ask the students to predict, we're at science practice 6 here also, predict the dry weights in increasing order. And this is a head scratcher for the students, right? Or sometimes maybe not a head scratcher, sometimes they pick the answer that they think it is, and they're not thinking through what is necessary to get to the right answer. Again, matter and energy are not interchangeable. The answer here is D, least amount of dry mass here, second, third. And then ask the students whether they got it right or wrong to justify their response. That's still at Science Practice 6. Justify your response. Why did you put what you thought, right? So this is getting at what? 2 processes, still with Science Practice 3 actually. What 2 processes are going on with light and water, no light and water? Well, actually only 2 processes are only going on with one of them. Photosynthesis and cellular respiration, right? So here, we've got photosynthesis and cellular respiration occurring simultaneously. And so as the cellular respiration is occurring, right, the cells are also photosynthesizing. And so asking the students something about, what's happening, what is adding the mass? Where's the mass coming from? Where is the mass coming from for a plant when they do photosynthesis? Where does the mass come from? What's adding the mass? Because the students will often say, it's coming from the soil. Nope, the new mass is not coming from the soil. It's coming from the carbon in the atmosphere, right? It's coming from the carbon dioxide. So these are putting on new mass while they're using the glucose that was stored in the seeds. These, on the other hand, are using the glucose that was stored in the seeds, but they're not putting on any new mass because they were in the dark and they're not doing photosynthesis, right? So and you can tell the students, they don't have to take your word for it. They can do it. This requires 3 Petri dishes or bowls. It doesn't even have to be a Petri dish. And some radish seeds, which are like 100 radish seeds for 32 cents. So this one is really easy to set up in your classroom and have the students collect the data on their own. I have a second visual of this. That shows another thing that you could do with this one. I love pulling data and having the students analyze the data. This is visualization, so it's Science Practice 2. This one is data analysis, so Science Practice 4. And you can ask the students to explain the change in mass of each of these investigative categories. So light and no water started out with 1.5 grams and ended up 1.49 grams, probably a little bit of user error there. The light and water started out at 1.5 grams and ended up at 1.63 grams. This is dry mass, by the way. And then this one started out at 1.5 grams and now is 1.21 grams. So then asking the students, why did that happen? Explain what happened, right? So here's our task verb over here, explain, and now they're taking this content and they're using it to answer a science practice question. This is a phenomenal lab. I do this one every year. I love it. This one is from this is from the College Board. This is an old FRQ here. You could use a lot of strategies with this one. Exit tickets, using assessment questions as a teaching tool. You could give the students the question. This is just a simple multiple choice question. If in Unit 2, does anybody do the dialysis tubing bag and they put the glucose and the starch? If you know what I'm talking about, you know what I'm talking about. This is a counterpart to that lab. But if you've never done the lab, there's enough stimulus here for the lab. But the question is asking the students, hey, which of these graphs, so that's Science Practice 4, which of these graphs correctly shows what would happen to the amount of, what are we looking at, water, starch, and glucose over the period of time from this setup to this setup? And the answer is A. The students will be able to recognize that if they can read graphs. But it's a nice practice. But then you can add to that. So this is what the question had, right? But then you can add to it. Describe what is happening in the graph that represents the correct answer, or explain why this is happening. These are all things that you can just add on from something that you got, from the College Board online, and having the students write it down. Here's the trick. Trick. Yeah, I guess it's trick. Here's the trick, is holding the students accountable for their own words. So what happens is, you guys probably know this already, the students want to know that their answer is right before they commit to it. You know, and that rarely happens in science, right? Like, that's just not how scientific investigation works. So holding their feet to the fire, making sure they're writing something down, even if they're just doing it for themselves, or they're sharing it with their seatmate, or sometimes I'll have them sketch stuff and I have them hold it up. Or if it's a multiple choice response, you know, having them hold up, you know, 1, 2, 3, or 4 fingers. Having them feel accountable so that then you get the idea of, like, who has a misconception here, or who didn't really get the content the first time. But then for questions like this, have them write it down, right? Like, having them be responsible for writing it down, I think, is a really important part, because even if they're orally answering a question is not the same feeling as having to write it down. And again, having graded at this point what feels like millions of AP FRQ responses at the end of the year, the students are just very tentative with the way they write and use a lot of, you know, like, it did this, and the reader doesn't know what they're talking about. You know, like, you have to be more, they need to be more articulate than that in their writing. So the second one is visual representations. This is hands down my favorite. This is my favorite one. I would put a visual representation every single day because I freaking love these. So this is, again, this is one that I pulled off an old FRQ. And I love these strategies. They're super quick, easy for the students to do in a minute or less, or talk to their partner about. If you show them a visual representation, this one happens to be a pathway, ask them to caption it, right? Caption this, and then they can share their captions, or somebody could raise their hand in if they want to, you know, contribute what their caption was. So, you know, don't put the caption on it. Snip that part out, and then have them caption it. Or you can remove the labels, right? Like, you can remove some of these labels, and you can ask the students, label this. So you can ask them to do that. You can ask them to describe the process that's happening from this visual representation. Or if you have one, let's see, explain the process that is occurring that upregulates the transcription of this target gene, and they can use the illustration to help them do that. So any of those things are phenomenal. Also, for visual representations, I have my students do something we call Sketch It, and they do it in their lab notebooks, but also sometimes on big giant paper. Full disclosure, these are not from my students. I have a colleague who was an art major and now is a science teacher. She did these, but I do have my students sketch a lot. So the nice thing about sketching is, especially if you're talking about something that is process oriented, so there's like 10 different steps to it, is that the students, when they get to a certain point, if they can't draw the picture, they don't know how to visually represent it, that's the clue for them. Oh, actually, I don't know what's happening right here, right? So the photosynthesis and cellular respiration are great examples of that. My kids don't do it up like this. It's like Christmas over here. But for example, if the students are writing about what is happening to, like within a thylakoid in photosynthesis, and they get to this part and they're like, wait a minute, I don't know what to draw after the arrow. I don't know what to draw as coming out of the thylakoids. What's coming out of the thylakoids going into the stroma? I'm not sure what that is. Well, that gives them a piece of information, right? That's some data that they need. Like, ooh, I thought I knew that content, but I don't know it. So this is really helpful for the students, but then it's also really helpful for me because, you know, they have a piece of, even if they're using like an 8.5 by 11 piece of paper, I can kind of walk around really quickly and I can see like, oh, they got stuck on this part or they moved past that part. That is something that is hard for me to get feedback on if they're just answering questions orally in class, because not everybody talks in the class during the class, right? But if you ask them to sketch something, you could do a poster session or a gallery walk or a 2-minute sketch or a 3-minute sketch. These are all things that you can incorporate every single day. Oh, the examples over here, cladogram, pedigree, what else do we have? Signal transduction pathways, everybody's favorite, Unit 4. This is an example from my good friend Jim Smanik. This is a 3D kinesthetic model where he had, it's a YouTube video, and if we were together for 2 minutes, I would play it through, but if you get this presentation, you can click on the link. It's a YouTube video. You can open it. Just to have some kind of three-dimensional model, right? Something that's in front of them. You can make it yourself. You can have the students make it. Jim made this one. These are little cups, like little medicine cups, and he's highlighting here chemiosmosis in cellular respiration, and he just cut out all of these little, they're like NADH, FADH, etc., and little hydrogen ions, and he's moving them across the membrane, and he's shuttling electrons using these little cups, these little medicine cups. Anyway, I think that's kind of cool. This is a really tough. I don't think I knew chemiosmosis until like 3 years into my teaching career, right? Like, this is something that's hard for the students. So, get yourself some little. Well, these are for making food webs, so that's not really translating to the chemiosmosis. But, get yourself some little illustrations and have the students literally move them around, right? When you're talking about hydrogen bonding, give them some little magnetic bits and pieces that they can utilize, not because they don't know what a hydrogen bond is, but because we want them to be able to articulate more than just a definition of hydrogen bonding, right? So this is great, this homemade modeling for the students. And I love this one too. So on the Tube of Views, you can find a video snip for pretty much any process that you were ever interested in, right? So you can pick up one that's like 1 minute or 2 minutes or 3 minutes, or you can, how you can like cut them down, you know what I'm talking about? Like you can cut them down to just the minutes that you want. And I have my students a lot of times watch, I think this might play, oh look, I'm like in my classroom. Oh my gosh, I'm in my classroom. I have my students watch these with no audio, and I have them narrate it, right? This is a great, like the kids love it when they didn't used to, but now they do. Love it when we do this, like can you narrate what is happening? Even if they're just narrating it to their seatmate or to their own brain. And you can have them do it in a couple of ways. They can share their narrations on an audio app if you're interested in that, like on their phones, you know what I'm talking about? Or they can just, like I said, narrate it to their seatmate. If instead of a narration, you're interested in having them describe the process or explain something about the process, that's also super cool. But I love that one because it's quick. You can find it really easily online, and it gives students an opportunity to use their own voice, which is critical. Metabolic pathways, again, show them a pathway, analyze this, describe this, explain this, sketch this. This is great. This particular illustration I love because it has an enzyme that has both an active site and an allosteric site. And this is a really good illustration of how the end product of a metabolic pathway can serve as an inhibitor of the enzyme. Again, this is something that the students really need practice with. You could do something like this using little cutouts, right, because they would be shape specific. That would be kind of cool. You could have your students draw them, or just give them something like this. Ask them to explain what is happening. Or shoot, you could just you could take off all of these labels and have the students label those. I don't know, I love that. And then here's another one, again, for metabolic reactions. You can ask them all kinds of questions about this. You could ask them to provide an example of something that you've talked about in class already that is catabolic. You could, you could show them this, and then you can have them draw like a reaction pathway graph, right? Like over time, like is this, if it's catabolic, right? What's the free energy of the reactants versus the products? Or if it's anabolic, what's the free energy? That incorporates graphing. So science practice 4. Love it. This might be the last one for visual representation. This one I really love. I asked my students to turn and talk this year is what I did with them in class this year. But I love this one because when we talk about the cell membrane, the plasma membrane, the students are like, I get it already. Stop, enough with this is hydrophobic and this is hydrophilic. The fact is, we tend to learn things the way that we see them, and then it's difficult to make a prediction about what would happen if something changed. And every single year on the AP exam, there will be a question where the students have to make a prediction about what would happen if something that they understand in science were changed slightly. There was a mutation in the DNA and now the enzyme doesn't have the proper configuration or whatever. So this is a really fun one. The SNP came from the College Board. I did not make that up. That's the SNP from the College Board. You were asking. A student proposes 6 models of a hypothetical plasma membrane. Okay, there's the 6 models over there. Describe the model that best represents the structure of a plasma membrane in a non-aqueous environment with the cytosol environment similar to living cells. I have never once given this to my students where wholesale every kid got it right. Never. Because they have like a search visual in their brain, right? What is the search visual for their phospholipid structure? It's a bilayer. The search image here is a bilayer, and the best response here is actually E. We have an aqueous cytosol. That's your content. That's part of your content. It just says what would it be like inside the cell? Aqueous. And a non-aqueous environment. It would look like this. That's the most reasonable, right? But having your students describe this or explain it is important. But that's a really good one to get the kids thinking about. That science practice 6 also, which is what if things were different? What if we didn't have an aqueous environment around all of our cells all of the time for every single cell on the planet? Then what would happen? Well, not what's happening right now. So this is a great way. And then you can think about other questions that maybe could be included with that image. You can probably think of many more. And then we talked about cladograms, asking the students not just, can you read the cladogram, which two share the more recent common ancestry. Those are the kinds of questions that typically would come up. But describe the characteristics of the cladogram, or explain the relationships between the organisms on the cladogram, or explain how this cladogram relates to larger biological principles. So what is the biological principle that's being highlighted in the cladogram? So those are all really good questions that could go along with this as well. What else do we have? This is a great one. So students can analyze a cladogram mostly pretty easily. However, when we ask them to draw a cladogram, which is something else that they have to be able to do on the AP exam, sometimes the wheels come off of the wagon. So can they take a data table and use the data in the table to come up with a cladogram? Now, this one is pretty simple. This is what I would start with my students with, because this one's pretty simple, because they know all of these things, and they know all of the derived characters that are listed, so they have a visual, right? They know what a mammal is, which one of these have hair, jaws, feathers, fins, whatever. They know that one, so this is where I would really scaffold this kind of question. So I would start with one that's like this. This is the expected response from that one. But then the next time I would give them organisms that they don't recognize, right? You could give them the scientific names of the organisms, or something like, I don't know, you could give them, organism number 1, 2, 3, 4, 5. You could even take for the characteristics for the derived characters, you know, character A, B, C, D, E, and then see if they can come up with this. Because if they can come up with this cladogram when they're talking about organisms that they don't know really, they haven't seen before, that's when they know how to do it. That's when they've got the analysis down, right? So that's a wonderful one, and every year for the past 3 years, the students have had cladograms on the AP exam. All right, so let's just look at this one for a second. I'm not going to, if you want the presentation, you can click on the link and you can look at all of these labs that I have on here. These are not all of the labs that I do, but these are some labs that really highlight Science Practice 3, which is, hey, do you know the difference between a null hypothesis and an alternate hypothesis? Do you know what the independent variable and the dependent variable are? I can 100% guarantee you they are going to have to know dependent and independent variables on their exam. What's the control group? Why did you pick that control group? Justify that. So all of these represent labs that do those things, but not just those things. They can gather statistical evidence using those. So one of my favorites are photosynthesis with the floating leaf discs. That's straight out of the College Board's lab manual. But in addition to that, M&Ms and chi-square analysis, right? It really just takes a bag of M&Ms, and you can do a chi-square analysis with an M&M bag. I like doing Simpson's Diversity Index, which is a new one that was added that when they redid the curriculum in 2018, the use of Simpson's Diversity Index. Love this one, gets us outside for a few minutes out of the day. We do Oreo double stuffs for statistical analysis. And the penny drop lab, that might be next. Let me see. No, it's not next. Let me see. There we go. The penny drop lab. The penny drop lab is the first lab that I do every year. And, I mean, you know the penny drop lab, like from when you're in second grade or whatever. But you can do a lot with the penny drop lab. You can, for 1 penny, the cost of 1 penny, well, I guess it's reusable, so not even the cost of a penny, the students can calculate the mean. That is one of the things, believe it or not, that they have to be able to do on the exam. And I don't know, for some reason in the science classroom, that doesn't feel the same as it does in a math class. So calculate the mean, explain why your results show something. For mine, I do penny drop 5 trials with water, 5 trials with water and soap, 5 trials with isopropyl. Right, and have them make a prediction at first, and then have them calculate the mean from those 3. I ask them to put that in a graph, to graph their mean data, which is also kind of fun because they have to be able to determine when it's appropriate to use a bar graph versus a line graph. They don't always know that. Like we need to practice that with them. So I have them do that. You know, ask them, hey, here's your hypothesis. What's the data going to look like if it supports your hypothesis, right? Or what's the data going to look like if it doesn't support your hypothesis? Ask them that ahead of time. And then if you click that link, it takes you to it. It's really lengthy. I have them do, this part's not, anyway, this is on the formula and equation sheet, but I have my kids do all of the statistical analysis with this penny drop lab, all of it. Like standard error of the mean, when they graph it, I have them include the error bars. This is a really low stakes way for them to practice that early in the school year, and then of course we practice it way later than that. But anyway, again, just trying to think of ways that every single day we can do something that's between 2 minutes and 10 minutes and 40 minutes that gives the students an opportunity to actually do something, like demonstrate what they can do and not just hear me talking about it.